Good morning, all. Myself Rajat HS from COH Kolar. We discuss about biofertilizers. Introduction. A biofertilizer is a substance which contains the living microorganism. When applied to the seed, plant surface or the soil, colonizes the rhizosphere or the interior of the plant and promotes the growth by increasing the supply or availability of primary nutrient to the host plant. Biofertilizers add nutrient through the natural processes of nitrogen fixation, solubilizing phosphorus, and stimulating plant growth through the synthesis of growth promoting substances. Types of biofertilizers: bacterial, fungal, algal, aquatic for earthworms. Bacterial fertilizers: the leaf cells of bacteria used used as a biofertilizer. These microbes contains the unique gene called as a knife gene, which makes them capable of fixing the nitrogen. The nitrogen fixing bacteria work under the two conditions: symbiotically free living bacteria, non-symbiotically. The symbiotic bacteria make the association with a crop plant through forming the nodules in their roots. The free living bacteria do not form any association but live freely and fix atmospheric nitrogen. Symbiotic nitrogen fixers. The most important symbiotic nitrogen fixing bacteria is Rhizobium and Azospirillum. Rhizobium living in the root hairs of the legume by the forming nodules. The plant roots supply the essential minerals and newly synthesized substance to the bacteria. The name Rhizobium was established by Frank in 1889. This genus has 7 distinct species based on cross inoculation ground concept. Azotobacter. Azotobacter is a heterophilic free-living nitrogen-fixing bacteria present in the alkaline and neutral soil. Azotobacter is the most commonly occurring species in the arable soil of India. Apart from its ability to fix the atmospheric nitrogen in soil, it can also synthesize the growth-promoting substances such as axin and gibberellins, and also to some extent the vitamins. The mass production of biofertilizers. WAM spores isolated, then spores mixed with the sterilized soil filled in the pots, the host plant transplanted in the pots. They kept 3 to 4 months in greenhouse. Soil in the pot along with the roots of host plant is macerated, dried till it attains 5% moisture. Dried soil inoculates used for field application. Azolla is a biofertilizer. Azolla is a tiny freshwater fern commonly common in ponds, ditches, and rice field. It has been used as a biofertilizer for a rice in all major rice growing countries including India, Thailand, Korea, Philippines, Brazil, and West Africa. The nitrogen fixing work is accomplished by the symbiotic relationship between the fern and BGA. Azole. In addition to the nitrogen, the decomposed azolla also provides potassium, phosphorus, zinc, and iron to the crop. Biofertilizer application method. There are 3 ways of using these nitrogen fixing bacteria: seed treatment, root dipping, soil application. The advantages of biofertilizers are: the renewable source of nutrients, sustain the soil health, supplement chemical fertilizers, replace 25 to 30% of chemical fertilizers, increases the grain yield by 10 to 40%, decomposes the plant residues, and stabilize CN ratio of soil. They improve the texture, structure, and water holding capacity of soil. No adverse effect on plant growth and soil fertility. Disadvantages: the biofertilizers require special care for long term storage because they are alive. The must be used before their expiry date. If other microorganism contaminated the carrier medium, or if growers use the wrong strains, they are not as effective. Biofertilizer lose their effectiveness if the soil is too hot or dry. Thank you.